Good afternoon, dear colleagues, members of the Gairdner Foundation and fellow laureates. I am truly honored to be the recipient of the 2021 Canada Gairdner Wightman Award. It is my privilege to speak with you this afternoon about my story of more than 35 years in clinical cancer research. I will show you how our work and that of many others have improved cancer outcomes, but also leave you with the recognition that remains much to be done. So to set some context, back in 1982, when I first began my job, here's what was happening in the world. The Canadian Constitution was being repatriated from Britain. The desktop computer was named the Machine of the Year. The acquired immunodeficiency syndrome was gaining traction in medical and research literature as a new disease. Michael Jackson released his Thriller album, and a young man from New York clearly already was interested in all things presidential. In July of that year, I had a 3-month-old daughter, Nicole, and a 2-year-old daughter, Danielle, and that's when I started my research work. But the story of cancer goes back much further. It's been part of the human condition for millennia, and as described in this lovely book by Siddhartha Mukherjee, one of the first documented cases of breast cancer was found in the Persian queen Atossa in about 500 BC. For hundreds or thousands of years afterwards, the early treatments of cancer were not terribly successful until the late 19th and early 20th century, when surgery was enabled by anesthesia and sterilization of instruments, and radiation, although discovered, was not used therapeutically until about 50 years later. Many patients had died within a few months or years of diagnosis. In Canada, in 1947, the Canadian Cancer Society created the National Cancer Institute of Canada to undertake research into the cause and treatment of cancer. 5-year survival rates at that time for all cancers were about 33%. In the 1940s to 80s, therapeutic radiation became possible with better understanding of physics, radiation dosing, and safety. New imaging technologies made it possible to stage cancers and thus plan treatment more accurately. And the first so-called chemotherapy drugs had been discovered, many through the US National Cancer Institute. By 1987, the first year for which online cancer survival statistics in Canada are still available, on the right of this slide, you will see that for some common solid tumors and leukemia, the 5-year net survival rates were well below 50%, with the exception of breast cancer. Big changes were afoot in cancer biology and clinical trials in the 1980s, however. In the laboratory, new techniques, technologies, and discovery and development of cancer models were used by scientists to begin to unmask the fundamental biological changes in cancer cells, foreshadowing new avenues for treatment in the decades to follow. In the clinic, Canada and many other nations created cancer clinical trials cooperative groups to conduct academic-driven multi-center studies intended to accelerate the pace of development of new effective treatments. In 1980, with funds from the Canadian Cancer Society, the National Cancer Institute of Canada founded a clinical trials group to coordinate, conduct cooperative clinical trials in Canada. Dr. Joseph Pater, my mentor and teacher at Queen's University, who was a hematologist and epidemiologist, was the first director of the NCIC-CTG. And in July 1982, I joined the NCIC-CTG as the first director of the IND, Investigational New Drug Program. The mission of the NCIC-CTG then, as it remains now, is to develop and conduct clinical trials aimed at improving treatment and prevention of cancer with the goal of reducing morbidity and mortality from this disease. To do this, 3 interlocking components are needed. Scientific and clinical leaders across the country needed to come together to develop the ideas that would be studied in clinical trials. Cancer centers and hospitals had to agree to become part of the group, since it was there that the patients who would be enrolled were treated. And a central statistical coordination center needed to be created to oversee the conduct and reporting and analysis of such studies. And all of these together formed the clinical trials group. In 2016, NCIC-CTG was renamed the Canadian Cancer Trials Group. In the beginning of the IND program, it's useful to reflect on where new cancer drugs were coming from at that time. Most of them were identified and developed by the US National Cancer Institute Developmental Therapeutics Program. Interestingly, and in contrast to today, pharmaceutical firms were generally uninterested in making a foray into cancer treatment because it was seen as too high risk with too low a return on investment. Canadian investigators and patients wanted the opportunity to be part of these early phase trials and to have access to promising new drugs. But these were awarded on the basis of letters of intent submitted to the NCI, and we were often too slow in Canada to get access. So in 1982, the Investigational New Drug Program was founded, and a junior oncologist, that would be me, hired as a director, where the role would be to identify new drugs in the NCI US pipeline in a timely fashion so Canada could prepare and be more competitive in accessing them. So useful to consider what I had as in terms of qualifications to lead a program like that. I, of course, had completed training in hematology. Oncology was not yet a specialty in Canada. I was once enrolled as a subject on a clinical trial, which is a longer story for another day. I played violin in the local symphony orchestra, so I did know something about collaboration and teamwork, but pretty much no other qualifications in terms of training, except I was keen and I very much wanted to be involved in clinical trials. On the other hand, at that time in Canada, there were few people indeed who had any experience and training in early cancer drug development. In the early years, the IND program began by focusing on phase 2 studies. These are trials designed to screen new drugs for evidence of activity in patients. Despite the fact that oncogenes had been first identified in the 70s and that would have an impact on future research, it remained the case that most new drugs that were coming out of the NCI US and elsewhere were cytotoxic agents coming from those screening platforms that I mentioned a moment ago. We did have a few lemons in the first years of the program, but also some exciting early results. Our phase 2 study of CBDCA, now called carboplatin in ovarian cancer, was the first in Canada and showed this drug was active in relapsed ovarian cancer. And now, of course, it plays a central role in the frontline and management of this disease. Interestingly, one of our lab scientists, a clinician named James Johnson in Winnipeg, had been doing some work in his lab on a rare leukemia called hairy cell leukemia and found a drug called deoxycoframicin seemed to be active. We were fortunate in being able to do a phase 2 study of this agent as well, and amazingly saw a 100% response rate with nearly 90% complete remissions in the patients we evaluated. The very first patient enrolled on this study was in Kingston by me. As a hematologist, I was accepting referrals of a variety of patient types. And fortunately for the gentleman who was referred to me at that time, we had just opened this study. So his new diagnosis of hairy cell leukemia allowed him to be entered. He had a massive spleen filling his left upper quadrant, going below his umbilicus. And after only 3 doses of therapy, he had a normal sized spleen and a marrow that was returning to normal. I can say there's nothing more exciting for me as an investigator, or frankly for the patient, than participating in research that has this kind of dramatic impact on a patient's life. The 90s were the decade of the taxanes. Taxol, which was its original generic name, but it later became paclitaxel, was an extract from the bark of the Western yew, a very complex molecule, as you can see here, and it was found active in the NCI screening programs in the 1960s. But it only became of interest decades later when Dr. Susan Horowitz, a 2019 winner of the Gairdner International Award, identified its novel mechanism of action as stabilizing microtubule formation. The early clinical trials in the 80s showed significant problems with cardiac toxicity, severe hypersensitivity reactions, perhaps due to the fact it was formulated in cremophore. But some evidence of activity in ovarian and breast cancer led to strategies such as pre-medication and prolonged infusion to try to overcome these problems. In Canada, the NCIC-CTG did a trial called OV9, which was my first foray into randomized ovarian trials. And I was interested in discovering whether better information about how to safely administer this drug could be obtained in a randomized design. So OV9 was the first CCTG-led international randomized trial in ovary. And what we did was we randomized patients to 2 different doses and 2 different schedules of paclitaxel in relapse disease. And I think surprising to all, we found that the shorter infusion was actually safer than a more prolonged infusion, and the higher dose was more effective. So this changed how this drug was given, not just in ovarian cancer, but in all other tumor sites, and remains to this day given this way. But it also did 2 other important things. The first was we developed a relationship with Europe and immediately moved paclitaxel into frontline trial with EORTC, where we showed that in the frontline setting in ovarian cancer, it had a fairly dramatic impact on overall survival with a 1-year gain. And again, paclitaxel in frontline ovarian cancer remains standard to this day. But it also created the idea that by collaborating across the ocean or between countries, we might undertake clinical trials faster. And that in turn led to the creation of the Gynecologic Cancer Intergroup, an organization of now 35 cooperative groups around the world, where meetings twice yearly identify interesting ideas for collaboration between groups, and periodic consensus conferences such as this one from 2004 in Baden-Baden identify standards of care and major questions to be addressed in the next few years. By the 90s, taxanes were everywhere. With the success of Taxol in ovarian cancer, dozens of trials were ongoing. And the first analog, docetaxel, emerged from a company in France with a theoretical advantage that it wasn't formulated in cremophore, so no pre-medication would be necessary. Because of the IND programs and the Canada's success with OV9, we were able to obtain this new agent for 5 phase 2 studies, including one in breast cancer, led by Maureen Trudeau in Toronto. And by the 10th patient, we knew 2 things with certainty. First of all, the drug was extremely active. 7 out of those first 10 patients had rapid tumor shrinkage and partial responses identified, but also that pre-medications sadly were needed because most of these patients had hypersensitivity reactions. And Taxotere or docetaxel went on to become part of the standard of care in breast cancer today. In the late 90s, changes in the air. Targeted drugs were emerging, new endpoints in trial design being discussed, and professionally, my life was also changing. I talk about the first new wave of targeted drugs, but it's important to remember that all cytotoxic chemotherapy agents do have molecular targets. But by this new wave, meant we were focusing for the first time on agents targeting signaling molecules and receptors thought to be the root cause of uncontrolled cancer growth. These pathways were very complex, as shown on the right in a yeast cell example. Nevertheless, there seemed to be some recurring themes on the types of receptors and pathways that were important in driving cancer. We expected these drugs to be less toxic, turn cancer into a chronic disease, and lend themselves easily to having a biomarker established to help us select which patients would benefit. In 1998, I was in Amsterdam at a new drug development meeting and was asked to give a lecture on my ideas about how these targeted drugs would change our clinical development pathway for cancer. The phase 1 endpoint for first in human trials, I believed, would be based on optimal target inhibition, not toxicity, and we could avoid that perhaps altogether. The phase 2 endpoint for screening for activity, we felt I felt targeted drugs would unlikely cause tumor shrinkage given their mechanism of action, so other measures would be needed. And of course, biomarkers for patient selection would be important and easy. Needless to say, we in the IND program began a large number of phase 1 and 2 trials of molecular targeted therapeutics over the next 5 to 7 years. And I list here some of the targets and agents that we studied. And most were negative, sadly, except for a proteasome inhibitor, bortezomib, in mantle cell lymphoma and Waldenstrom's, and a RAF inhibitor, sorafenib, in AML. That being said, the clinical trials group as a whole did conduct phase 3 trials with several agents, some of which improved survival in colorectal cancer and non-small cell lung cancer. Both of these were EGFR inhibitors, and the CTG studies led to initial approval of these drugs for their use in treating these diseases. By 2005, the targeted therapy era was being viewed as a somewhat mixed response. We did see some striking and game-changing results, such as imatinib in chronic myeloid leukemia, some less visually dramatic but important and practice-changing results, such as the HER2 inhibitor trastuzumab given in adjuvant breast cancer. But many were negative, or even worse than negative, with poorer results in the experimental arm. So the bloom on the rose of targeted therapy had begun to fade slightly, or maybe it was just that realism was beginning to set in. Not everything was going to be in imatinib in chronic myeloid leukemia or in GIST. My 1998 predictions, meanwhile, were actually often wrong. These drugs were toxic. Toxicities were different than chemotherapy, but they were not always easy to give. Dosing based on target inhibition was a challenge and sometimes actually wrong. Targeted drugs actually did cause tumor shrinkage. And in fact, in a review published a few years ago by myself and one of my fellows, we showed that those targeted drugs which had the greatest degree of tumor shrinkage observed in phase 2 were the most likely to have an impact on survival in phase 3 trials. And finally, biomarkers were definitely important, but they weren't anywhere near as easy to identify and study as we had thought. So this is a humbling reminder that those predictions I made based on what we knew from the lab needed to be evaluated in the clinical trials setting, and our knowledge of biology from the lab may be proven wrong when we actually take it forward into the clinic. As this lesson was unfolding, professionally, I was moving in a number of other directions. Increasingly, I was getting involved in cancer research organization leadership, in teaching about clinical research. And sometimes I was asked, like, why are you doing this? You're busy enough already. And I think there were a few reasons. Firstly, clinicians do add value to discussions about the directions of cancer research because we're familiar with the clinical problems and the day-to-day reality of cancer treatment. Secondly, I think we need to give back, being the recipient of funds from charitable and other organizations. Helping them achieve their mission through getting involved is important, as is teaching the next generation of clinical trialists, in this case, at a FLIMS meeting in Switzerland on clinical research methodology. And finally, because on a personal level, it was fulfilling and fun, and I met lots of new colleagues who became friends and continue to be so. In the next 5 years, between 2005 and 2010, therapeutic options continued to evolve and grow exponentially. The genomic revolution and rise of precision medicine started to take root, and anti-angiogenesis drugs were emerging. And professionally, I was moving more into the sphere of cancer control and policy. The brilliant Dr. Judah Folkman outlined the role of angiogenesis in promoting and sustaining tumor growth, as well as identifying inhibitors. And expectations were high when one of the first of these drugs, endostatin, went into clinical trials, and words such as cure were whispered in the hallways. Other agents targeting vascular endothelial growth factor, VEGF, or its pathway were being developed as well. Needless to say, the IND program was very involved. We were very involved in studies of these agents. We did several phase 1-2 trials. And I'm just going to highlight one here with cediranib, a VEGF receptor tyrosine kinase inhibitor. And our phase 1-2 studies led to phase 3 trials in both lung cancer and ovary. And while the lung study was negative, the ovarian study, which was designed with and led by our UK partners through the GCIG, did show a significant improvement in progression-free survival. But as shown here, although the overall survival data were intriguing, this did not quite reach the mark of statistical significance. The anti-angiogenesis wave of trials became the largest we've ever seen. Pharma was now firmly involved in cancer drug development, and multiple analogs and versions of the VEGF receptor tyrosine kinase inhibitors, as well as VEGF inhibitors, were available. And more than 1,000 trials of VEGF inhibitor bevacizumab alone at one point were open or completed. Despite all of these studies, angiogenesis inhibitors today have only rarely had a meaningful impact on cancer survival. And there's some lessons here. The first is around the preclinical models we used to get excited about this class of agents. Preclinical models, as shown in this fairly old illustration from Ian Tannock, have extremely rapid doubling times compared to, in this example, human breast cancer. So it's not surprising they're highly reliant on neoangiogenesis. And the second lesson is that when a drug or drug class has limited activity, you cannot make it active by doing thousands of clinical trials. As I said a moment ago, I became involved more in cancer research policy, and really my main focus was the Canadian Cancer Research Alliance. Now, this organization was formed by Drs. Phil Branton and Vic Ling back in 2003, when 20 agencies involved in cancer research funding came together to share their information on what they were investing in and strategy. And by 2021, this organization, which is still growing strong, has 33 cancer research funders, and they're investing close to $500 million in cancer research in Canada over the years. I co-chaired the CCRA, which was supported by the Canadian Partnership Against Cancer for nearly a decade. And in that time, one of the questions we thought was important to ask was, how do we collaborate better between agencies and through shared investment accelerate impact? And with that kind of thinking and a lot of work, we put together the first Pan-Canadian cancer research strategy. There were 24 action items outlined in those strategy, 22 of which we were successful completing. But one explored something very dear to my heart, which was the state of academic cancer trials, which were really struggling at that time. One of the recommendations of that report was to create a Canadian Cancer Trials Network, which would supply infrastructure support to hospitals and cancer center to enable academic trials to take place. And we've done that. A multi-agency funded initiative led by Janet Dancy and housed at OICR has led to a 200% increase in academic trial enrollment in the last 5 or 6 years. Moving to our current decade, we are, of course, in the time of the immune oncology revolution. Professionally, a number of changes in my life were taking place. Let's first talk about the immune therapy. I was going to say wave, but it is more like a tsunami. Immune checkpoint inhibitors, CTLA-4 and PD-1, PD-L1 inhibitors have really been transformative, in particular to melanoma, but active in other cancers. CAR T cells, engineered T cells have shown activity in hematologic malignancies and likely will as well in solid tumors, which are undergoing research. And in the CCTG, numerous clinical trials are ongoing or have been completed. I was involved in helping to design and get started an adjuvant lung cancer study, an international study, which we hope will be analyzed soon. And you can see some of the other studies here. And recently, with federal funding, a CAR-T platform has been set up with Annette Hayes' leadership at CCTG. But important to reflect too, this is a tsunami. By a year ago now, there were actually 4,400 clinical trials of PD-1, PD-L1 inhibitors, reflective not of 4,400 good ideas, because there are multiple such inhibitors, and many of these are actually identically designed or extremely similar studies. So while this is exciting, it's also concerning that many of these trials, being identical and similar, if they fail, will amplify failure and waste resource. In 2012, a shift began in my life. I was department head in oncology at Queen's, and I attended a World Cancer Congress where Chris Wild, head of IARC, said that the global cancer burden is or will become so large we will not be able to treat our way out of the cancer problem. And as someone who'd spent her whole life investigating cancer treatment, this was something new to reflect on, and it increased my personal interest in prevention and palliation. So just 2 words on those. Tobacco, which is the most common preventable cause of cancer. I got active in this area, worked with a team to develop a national summit to create an end game for tobacco. And our end game goal of less than 5% tobacco use by 2035 was adopted in 2017 by the federal government. In terms of palliative care, of course, as an oncologist, I have considerable experience with the need for optimizing palliative care. And despite this, it was clear that it was hard to ensure it was well integrated and available not just to all cancer patients, but to all patients, period. Now, discussions around the dinner table on this subject were important because my husband, a family physician who did a lot of palliative care, and my daughter, Danielle, now a palliative medicine specialist at Queen's, got me extremely vocal about this area. And in a few years ago, Danielle and I actually co-wrote our first, hopefully not last, paper about the integration of palliative care and how we can optimize it into oncology care. So let's circle back to say, what have we actually done? What has happened in the 35 years or more since I started my career? Well, first of all, 5-year net survival is now 63% in Canada. And those 4 solid tumor types I mentioned way back at the beginning of my talk, you can see that all of them have improved dramatically, with maybe the exception of lung cancer. And while some of this may be due to early detection, it's clear looking at mortality data that it's also due to better treatments. We still have a long way to go for some cancers, but we have shown improvements in others, and CCTG and the work we did has improved, has contributed. So in 35 years in clinical cancer therapeutic research, we've evaluated cytotoxics, targeted drugs. We've had waves of taxanes, angiogenesis inhibitors, and immune agents. So what's next? Well, it's clear cancer biology and discovery research will continue to unveil new opportunities for therapy. Data is an all-consuming discussion in probably every cancer center. We have mountains of it from patients and biospecimens, imaging, and it's being harnessed to understand predictive and prognostic indicators and to design new treatments and drugs. My advice to the next generation of clinical cancer researchers is shown here. Be collaborative, resist the push to endlessly duplicate research, and engage patients as partners, which was a really important aspect of my own work. My advice to cancer research funding agencies: invest in academic clinical research, create the infrastructure for healthy clinical trials community so trials can be conducted beyond those done by pharma. And this applies more broadly than just for cancer research. So I'd like to thank the many organizations, in particular the Canadian Cancer Society, that funded the research work we did over the many years, and also the many mentors, colleagues, trainees, and literally thousands of patient volunteers in the trials and in our committees who helped design them. I'd especially like to thank my family who've supported me unfailingly. My family's grown larger, grown up, and definitely grown older. It has been such a privilege to work in these times. The CCTG has made a difference here to patients in Canada and around the world, but our work is far from done. Too many people are still developing, suffering with, and dying of cancer. We have promises to keep and miles to go before we sleep. And with that, I will acknowledge those lovely words at the end of this Robert Frost poem, and thank you for your attention.